Diabetes drug shelters pancreas cells from immune system attack. Scientists at Johns Hopkins Medicine say that an experimental monoclonal antibody drug called MAP43 appears to prevent and reverse the onset of clinical type 1 diabetes in mice, and in some cases, to lengthen the animal's lifespan. The drug is unique, according to the researchers, because it targets insulin-making beta cells in the pancreas directly and is designed to shield those cells from attacks by the body's own immune system cells. The drug's specificity for such cells may enable long-term use in humans with few side effects, say the researchers. Monoclonal antibodies are made by cloning, or making identical replicas of, an animal including human cell line. The findings, reported online recently and in the May issue of Diabetes, raise the possibility of a new drug for type 1 diabetes, an autoimmune condition that affects about 2 million American children and adults and has no cure or means of prevention. Unlike type 2 diabetes, in which the pancreas makes too little insulin, in type 1 diabetes, the pancreas makes no insulin because the immune system attacks the pancreatic cells that make it. The lack of insulin interferes with the body's ability to regulate blood sugar levels. People with type 1 diabetes face lifelong injections of insulin and many complications, including stroke and eyesight problems if the condition is not managed properly, says Dax Fu, Ph.D., Associate Professor of Physiology at the Johns Hopkins University School of Medicine and leader of the research team. Fu says MAP43 binds to a small protein on the surface of beta cells, which dwell in clusters called islets. The drug was designed to provide a kind of shield or cloak to hide beta cells from immune system cells that attack them as invaders. The researchers used a mouse version of the monoclonal antibody and will need to develop a humanized version for studies in people. For the current study, the researchers gave 60 for non-obese mice bred to develop type 1 diabetes a weekly dose of MAB43 via intravenous injection when they were 10 weeks old. After 35 weeks, all mice were non-diabetic. One of the mice developed diabetes for a period of time, but it recovered at 35 weeks, and that mouse had early signs of diabetes before the antibody was administered. Scientists say that an experimental monoclonal antibody drug called MAP43 appears to prevent and reverse the onset of clinical type 1 diabetes in mice, and in some cases, to lengthen the animal's lifespan.